Hello, everyone. I'm Chen Ji. Today, we'll talk about the notch signaling pathway, including the history, the structure, the function, essential. As you know, the notch signaling is an evolutionarily conserved intracellular signaling which is essential for proper embryonic development in organisms as diverse as insects, nematodes, echinoderms, and mammals. Notch receptors initiate a highly conserved signaling pathway that influences cell fertilizations within multiple tissues and regulate the ability of precursor cells to respond to other developmental signals. In mammals, notch signaling regulates neurogenesis, myogenesis, vasculogenesis, hematopoiesis, skin development, and other aspects of organogenesis. In addition, notch signaling is involved in other critical cellular processes such as proliferation and also apoptosis. Consistent with the ability to influence cellular differentiation in multiple tissues, mutations of notch receptors and components of its signaling pathway have been associated with a number of diseases, including human T-cell leukemia. Firstly, let's take a look at the history of notch signaling. The notch gene was first described in 1917 by American Joyce of genetics and embryology. His name is Thomas Hunt Morgan. He found a strain of fruit fly, Drosophila melanogaster, has notches apparent in their wings. The fruit fly has only a single identified notch-like gene, which defects resemble the typical notch mutant phenotypes. Its molecular analysis and sequencing was independently undertaken in the 1980s by Sperus A.T. and Mitchell W.Y. Sperus showed primary structure of the major embryonic notch transcript by sequence analysis of overlapping cDNA clones. The 10,148 BP sequence corresponding to this transcript possesses an 8,109 BP open reading frame that potentially codes for a 2,703 amino acid protein. They also found that this polypeptide contains a repeated structure composed of 36 randomly arranged 40 amino acids long repeats, which show homology to the epidermal growth factor and other proteins containing EGF-like repeats. Hydroxy plots suggest that the putative notch protein may spine the membrane. In 1991, Ellison Essential identified a translocation of chromosomes 7 and 9 in T acute lymphoblastic leukemia patients, which resulted in fusion of the thread region of Notch1 into the TCR locus and consequent overexpression of the active form of Notch1. The earliest evidence for non-canonical notch signaling came from in vitro studies, in which increased Notch1 level inhibited the differentiation of myoblast cells into muscle cells. The authors reported that the inhibition of myoblast differentiation did not require the CSL interacting domain of Notch1 and was not mediated by CSL or known Notch target gene, suggesting the existence of CSL-independent Notch pathway. This finding provided compelling evidence that ligand CSL-independent function of Notch is present and active during development. We call it a non-canonical Notch. Let's take a look at the structure of Notch signaling. Canonical Notch includes Notch receptor, Notch ligands, and target genes. There are 4 Notch receptors, including Notch1, Notch2, Notch3, and Notch4. 5 of Notch ligands include JAK1, JAK2, DELTA-like1, DELTA-like2, and DELTA-like4. The target gene are the HERS and HEY gene. Mature Notch proteins reside on the cell surface as non-covalently linked heterodimers that are comprised of extracellular and transmembrane Notch polypeptides. Extracellular Notch proteins are characterized by numerous epidermal growth factors. We call it EGF-like repeats. Transmembrane Notch1 and Notch2 are almost identical in size and share many structural features, including the membrane proximal RBPG associated molecular, we call it RAM domain, which mediates interaction with several cytosolic and nuclear proteins. The ankyrin, we call it the ANK domain, which is important for protein-protein interactions. 2 nuclear localization sequences, we call it NLS. A carboxy terminal transactivation domain, TAD, which is important for activating transcription. And the past, we call it a protein glutamate, serine, and serine-rich domain, which is important for regulating degradation. Transmembrane Notch3 and Notch4 are shorter and lack the TAD. The heterodimerization domain, we call it a HD, spans the region of interaction between the extracellular and transmembrane portions. Mutations in this region result in ligand-independent activation of Notch receptors. There are 5 mammalian ligands for canonical Notch pathway. Each ligand contains an EGF-like repeat region and a conserved sequence that is also found in Drosophila melanogaster, Cynohepatitis elegans, and vertebrates, and is known as delta serrate lac. We call it a DSL. The Notch receptor is produced in the endoplasmic reticulum and processed by furin-like convertase. It is a S1 cleavage in the Golgi compartments. This S1 cleavage is necessary for cell-phase expression of Notch, but is not directly involved in the ligand-induced release of active intracellular domain. At the cell surface, the Notch receptor interacts with the Notch ligand presented on the juxtaposed cell. After ligand interaction, processing by ADAM, metalloprotease, this is a S2 cleavage. The receptor is proteolytically cleaved by the gamma secretase complex. This S3 cleavage, which can occur at the cell membrane or in endosomes, here depicted only at the cell surface. The architecture of the gamma secretase complex is seen in the picture. In picture A, subunit topology, gamma secretase consists of 4 proteins: PSEN, NACT, alpha-1, and PEN2. PSEN forms a catalytic subunit. This is a catalytic aspartyl residues are indicated with stars. The endoproteolytic cleavage site in the cytoplasmic loop of PSEN is indicated. Stars on the luminal domain of NACT indicates complex glycosylation of the mature form of NCD. B. Schematic representation of the 4 different gamma secretase complexes. The heterogeneity is based on existence of 2 different PSEN genes, PSEN1 and PSEN2, and 2 different alpha-1 genes, alpha-1A and alpha-1B. We can see it in the color insert. Then, the Notch ICD is liberated and is translocated to the cell nucleus. Alternatively, the receptor can also be degraded upon relocation to the lysosome. Notch ICD forms are trimeric complex with CSL and MAML, which converts CSL from a repressor to an activator and initiates transcription of Notch downstream target genes. The best characterized Notch targets are the BHLH genes of the HERSH families. HERSH have now been shown to function downstream of Notch in many critical processes, including proliferation, differentiation, apoptosis, self-renewal, and asymmetric cell division regulation, and to contribute to oncogenesis. Apart from the HACE and HAY genes, the Notch-related anchor repeats proteins, CMYC and delta-like, are regulated by Notch. For example, the MYC gene is a direct target of Notch in several types of cancer cells. Knockdown of MYC in these contexts compromise the extent of proliferation, arguing that the MYC is the important intermediate in the proliferative response to Notch activation. Other direct targets involved in promoting proliferation, including cyclin D, string, CDC25, and CDK5. ICN, that's an intracellular Notch activating complex, is short-lived, and ICN gets photorylated on its past domain and subsequently ubiquitously by FBW7 and targeted for degradation by the proteasome. This is the components and activation of canonical Notch signaling. Non-canonical Notch signaling is CSL independent and can be either ligand dependent or independent. Some of the non-canonical Notch ligands include CCN3, microfiber-associated glycoprotein family with MAGP1, MAGP2, F3 contactin 1, MB3 contactin 6. Although some genes, including F-kappa B, GNK, or ROS essential, are affected by non-canonical Notch function. In most cases, the mediators of large canonical Notch signaling are unknown. The most well-studied and conserved effect of non-canonical Notch function is regulation of Wnt beta-catenin signaling. In the presence of Wnt, membrane-bound Notch forms a complex with active beta-catenin and degrades activate beta-catenin through an endolysosome pathway. Notch ICD can also regulate active beta-catenin levels in a similar mechanism, but it is unknown whether endogenously processed NICD regulate active beta-catenin proteins. Notch bands and titrates levels of the obligate Wnt signaling components, active beta-catenin. Therefore, active beta-catenin activity may serve as a useful readout for non-canonical Notch signal. Next class, we'll discuss the Notch signaling pathway in hematopoiesis. Thank you very much. Thank you.